All right. Hi and welcome everyone. Thank you for joining today's The Plant Cell Focus Issue webinar on the subject of proteolysis. My name is Jason Padilla and I am your technical host for today's webinar. Today's webinar features three speakers, Marcela Rojas-Pierce, Leila Feiz, and Jiorgos Kourelis. The webinar is hosted by the Plant Cell editor, Ari Sadanandom, and is moderated by the Plant Cell Features editor, Margot Raffeiner. She will be moderating and handling all the questions later on. Next slide. The focus issue, as you know, is now available online, and we'll hear more about all the content in just a few moments. Next slide. This webinar is recorded and will be posted on our YouTube channel. Be sure to check those out and find other previously recorded the plant cell and the plant physiology webinar if you're looking for additional webinar contents surrounding the plant cell and plant physiology contents. Next slide. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB member dues help support making this webinars possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10 to receive a discount on your registration. ASPB members get early access to these webinars and additional great benefits, and you can learn more about ASPB and the other opportunities we provide at aspb.org. Next slide. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your attendance today. If you are experiencing technical issues, you can let us know about those using the chat box or emailing me directly at jpadilla@aspb.org. If you have questions for our guest speakers, please add them to the Zoom Q&A box. Our moderator will be sharing them with our presenters during Q&A. If you are having technical difficulties or having issue connecting or need to leave early, this webinar is recorded and will be made available later on. Be sure to follow the Plante YouTube channel to receive notification when new content is posted. All right, so with that, I'm going to turn it over to our editors who will kick off the webinar and tell us more about the focus issue. Take it away, Margot and Ari.
Thank you, Jason. Hi, Margot. Thanks very much for moving the slide on. Hello, everyone. My name is Ari Sadanandom. I'm one of the reviewing editors of Plant Cell, and I worked on the focus issue. And today's webinar highlights authors from the current Plant Cell focus issue on proteolysis. And I'd just like to give a short introduction to this focus issue. So proteolysis is an essential cellular function, not only for protein turnover and removal, damage to unwanted proteins, but also for altering protein function, including protein-protein interaction or enzyme activities. This is to ensure there's optimal protein stoichiometries in the cell as the plant cell is responding to the environment or intrinsic or extrinsic cues for growth and development. And of course, you would know that proteolysis plays a central process in hormone signaling, defense against pests and pathogens, and abiotic stresses, and also is critical in basic cellular functions like cell cycle, metabolism, organelle biogenesis, maintenance, and even senescence. Now, what many people don't realize is that compared to animals, the plant genomes encode a highly expanded number of components related to proteolytic or proteostasis pathways. For example, the ubiquitin E3 ligases in plants, the number in plants, for Arabidopsis, for example, is 10 times larger than in humans. And so therefore, it kind of shows the reliance of proteolysis or protein turnover as a key mechanism for driving growth and development in plants. This is also true for autophagy, endosomal trafficking, and organelle-associated protein degradation pathways. Next slide, please, Margot. So the article in this focus issue contributes a wealth of new information and insightful review and discussion of the state of the art of proteolysis research in plants. And we hope that this focus issue stimulates further progress in this area. And that's one of the key drivers for getting this focus issue together. I've listed some of the papers here with in-depth reviews Could you move the next slide as well, please? And also, 3 articles we're highlighting where they will give a short presentation on this work. And not only that, the issue also has commentary by experts and proteolysis who pose and discuss compelling questions in these areas of research. For example, we have 2 letters to the editor addressing the question of whether the polyubiquitin pathway operates inside intact chloroplasts. The question is posed by Van Wijk and Adam, with a reply from Jarvis et al. Both make very valid points on the methodology used and how they analyze the data coming through. And we eagerly await more definitive data to arrive at a complete understanding of how protein degradation occurs inside the chloroplast. So without further ado, I'm going to pass you on to Margot, who's going to moderate the sessions. Thank you for listening. Hope you enjoyed the webinar.
Yeah, thank you very much for the introduction. So my name is Margot. Hi, everyone. I'm very happy to moderate this webinar where we are able to highlight some of the research papers that have recently published in the new Focus issue about proteolysis. And it's my pleasure to moderate this webinar. And so I will shortly introduce each of the speakers today. And afterwards, after each talk, we will have the opportunity to ask some questions. So please, every question, as Jason just said, text them or write them in the Q&A box, and I'll try to read them and present them to the authors. So let's kick off this nice webinar by introducing the first speaker, which is Professor Marcela Rojas-Pierce. Sorry for the pronunciation. So she did her PhD in botany at UC Riverside, but is now a professor in the Department of Plant and Microbial Biology at the North Carolina State University. And her lab is basically interested in the regulatory mechanisms behind vacuole biogenesis and also vacuole dynamics. But she's also interested in the transport of tonoplast proteins to the vacuolar membrane. And today she will present us a tool that was developed to study the rapid depletion of target proteins in plants that could be used to elucidate the function of very essential proteins. So we are very much looking forward to your talk, and the stage is yours.
Thank you, Margot. Let me see here. Okay. All right. Thank you so much, Margot, and the editors for the invitation to present our work. I want to start by giving a shout out to Dr. Liwen Jiang, who's the first author on this paper. He unfortunately he couldn't give this talk. He's now an assistant professor at Zhejiang A&F University. So this is not working. Okay, so my lab, let me turn this off. Okay, so my lab studies very dynamic processes, including vacuole fusion in stomata and in other cells. And these are cellular events that are really fast. And many of the genes that are involved in our pathways are typically essential. So we don't have access to mutants because they're all embryo lethal. And so the only tools that we have to study the proteins that we want to study are like inducible silencing mechanisms or silencing systems that take days for us to see phenotypes. So this sort of inspired our interest in developing a tool that allows us to degrade proteins at a specific time in plants that have already either matured or developed to overcome these challenges. And so we got interested in this system, after seeing the success of similar degron systems, one of the most famous one is the auxin-inducible degron. And this is a system to target proteins for degradation that takes advantage of the auxin pathway. So in the auxin degron, people fused a degron which is based from an AUX-AAA protein. These are the transcriptional repressors. And they co-express this fusion of the protein of interest to the degron with the expression of the tier 1 receptor from plants, such that when you have these proteins expressed and you apply auxin, auxin induces the recruitment of the target protein to this now chimeric E3 ligase that contains tier 1. And this results in ubiquitination of the target and degradation by the proteasome. And so this is the original data from yeast cells that were used to develop this system. So these are cells that have been transformed with tier 1 and this aid auxin degron fused to GFP. And what you can see here is that within 30 minutes, you can see degradation of GFP in these cells. So we were really interested in being able to do these kinds of experiments in plants, but obviously we cannot use auxin because auxin is such an important regulator of development. One example that has been used in plants a couple of times is the degrade FP. This is another protein that takes advantage of an F-box protein. In this case, the F-box protein is from Drosophila, but it also associates to form a larger chimeric E3 ligase to degrade targets. The way that they recruit targets in this case is via a nanobody that recognizes GFP, which would recruit then this protein of interest to the E3 ligase, again, for degradation. This has been used in plants a couple of times. In particular, it was used to degrade Wuchel GFP fusion in the shoot apical meristem. And this was using an inducible system. I believe it was the alcohol inducible system. So having these examples really pushed us to think about how do we develop a robust protein degradation system in plants that could be applied to many contexts. And so we looked for E3 ligases or an E3 ligase substrate pairs that would be identified from non-plant systems to ensure that we had fewer possibilities of targeting endogenous proteins. And so this protein, the Salmonella SSPH1, was a really, really interesting candidate for our work. So SSPH1 is a protein effector from the bacteria. It's released into human cells. It targets a protein kinase in humans called PKA1. And the enzyme is an E3 ligase that contains both catalytic domain here, shown in this globular domain. And this catalytic domain absolutely requires the cysteine 492. So if we mutate cysteine 492, you generate a non-functional enzyme. SSPH1 also contains a target binding domain, which forms a leucine-rich repeat structure shown here. And so the nice thing about this is that this enzyme then combines both ubiquitination and substrate binding in the same protein. The other major reason why we selected this enzyme is that the interaction between that leucine-rich repeat domain and the target was really well known. So there was already a crystal structure that identified the residues that are involved in interaction between this LLR domain and what's called the HR1b domain of the substrate. This is a helix bundle that binds into the surface of the leucine-rich repeat. And so having that information allows us to really hone in and design the minimal fragments that are necessary to bring these two proteins together and to ubiquitinate the target. And so and from prior work, it was known that this LR fragment, the fragment that contained this leucine-rich repeat in the catalytic domain, were sufficient for substrate recognition and ubiquitination. And on the side of the substrate, it was known then that HR1b domain, which is only this 78 amino acid helix bundle here, were sufficient again to recruit a substrate to the enzyme. And as I said before, because it was known how this enzyme worked, it was very easy to develop negative controls, including a catalytically inactive enzyme, as well as a substrate or a degrant domain that did not bind to the enzyme. So if we look at it in cartoon form, this is the endogenous or the native protein. So this is the substrate PKA1, and we represent the degrant domain here as HR1b. And then the Salmonella SSPH1 enzyme contains, again, the catalytic domain, the E3 ligase domain, as well as the leucine-rich repeat domain. And when these two are found together, this results in rapid ubiquitination of PKN1. So in our system, we would use the HR1b domain as a degrant, infuse it to a target of interest to allow the recruitment to what we now call E3 DART, which is this fragment that contains only the leucine-rich repeat and the E3 ligase domain. And these would allow to then promote the ubiquitination of the target protein. So first, we had to test whether this enzyme worked correctly in plants. And we used that in Nicotiana transient assays by co-expression of E3 DART, which is always fused to M. cherry so that we can visualize its abundance. And this was expressed together with HR1b or the degrant fused to a GFP as a proxy for target abundance. So when we co-express these two proteins together, we always saw very, very little levels of GFP, suggesting that E3 DART was in fact active and was able to ubiquitinate and promote degradation of those HR1b targets. And here's our negative control. If you mutate the residues that are important for that interaction, this protein is insensitive to the E3 ligase. And similarly, if we use a mutant form of the ligase, which is catalytically inactive, then degrant tagged GFP is again insensitive. So indicating that this interaction is specific and that the ubiquitination activity is very robust. And we use immunoblots to show that these low levels of GFP are really due to protein degradation, because when we co-express these two proteins, again, we see very little, no levels of the GFP protein by itself. So the system worked really well in transient assays. We wanted to develop a system that we could actually induce. And I encourage you to read parts of the paper where we describe things that didn't work to induce protein degradation. But we decided to make it inducible by using a traditional DEX inducible system. And so here's, this is a transcriptional induction of E3 DART based on the GVG system. So in the absence of dexamethasone, the enzyme is not expressed, but if we add dexamethasone, now the transcription factor here is able to activate transcription. The enzyme is expressed, and now it should be able to degrade this target. So we showed that we can actually induce expression of E3 DART under these systems when we apply dexamethasone, and the system is not leaky. So in the absence of dexamethasone, there's no expression of E3 DART, so that was nice. And you can see that here also if you look at fluorescence microscopy. But most importantly, we actually showed that when we induce the expression of E3 DART, we actually see decrease in the levels of that HR1b GFP substrate. And so the system then, the enzyme, the abundance of the E3 DART ligase results in the degradation of HR1B, which was very, very exciting for us. And in Nicotiana, we could see an effect after 3 and 6 hours of induction. We showed that it was also true for fusions to other proteins, including BPS34. This is a very large protein in plants, as well as RDH6, which is a very small transcription factor. And so this protein is in the nucleus, BPS34 is in the cytosol. So this adds flexibility to our system. So we think that many proteins can be tagged with the degron to induce degradation by E3 DART. Now, I do want to add a caution statement here, and that is that when we tested this in Arabidopsis, we did not see this beautiful induction. And so we tested the same GVG induction system, and we did not see we didn't see expression of the enzyme in Arabidopsis. And that could be due by many things that I don't have time to discuss. I'll be happy to answer questions. But nonetheless, we think that E3 DART it's a good system to apply to plants because it has low levels of probability of targeting endogenous proteins. We showed that it was very robust in degradation of HR1B labeled targets. And we think that it can be used, for example, if you express E3 DART with a tissue-specific promoter, so degrade your protein of interest and develop specific developmental stages. It could be used with other inducible systems that may be more robust. So in our hands, the GVG system was not great. We're also working on post-translational control of E3 DART, and that would be discussed another time. I want you to check out Adgene. We posted a bunch of plasmids in Adgene to share with the community, and we hope that people really adopt it. We are extremely grateful to NSF for funding. This was funded by an EGOR grant from the NCB program, and to Jose and Ana Stepanova's lab, who really got us into golden break cloning, which we could not have done it without it. And again, Lingzhu Huang was the main author on this paper. And I'll stop here because I think I went over time.
All right. Thank you very much, Marcela, for your interesting talk. So yes, you're a little bit over time. That's why Q&A box, if you can still hear me, I hope. So we have a couple of technical questions, actually. So there is one question, if the strain of Salmonella is not pathogenic, and how you check this?
If the strain of Salmonella
if the strain is not pathogenic, yes.
So we don't work, that Salmonella originally, yes, it's pathogenic, but we're also only working with a small fragment of the protein, so we never work with the, but yes, this is an effector that the Salmonella would produce during infection.
Okay. Thank you very much. So there are from the same person, there are two more technical questions. So maybe you want to contact Marcela afterwards. I think she would be happy to share it's more about microscopy techniques and cherry sources. So I think, yeah, maybe you can contact the author directly. And maybe as a last question, so generally there could be a leaky expression using the DEX system. Can be there any leaky expression? Can you answer this?
In Nicotiana, we did not see leaky expression as detected by fluorescence and immunoblots. In Arabidopsis, no. In Arabidopsis, our issue was that there was almost no expression whatsoever of the ligase. That was our problem.
Okay. Good. Thank you very much again for this very interesting talk. And I would like to move on to the second speaker of today's webinar. So I would like to welcome Dr. Leila Feiz. So you could already start your screen sharing while I introduce you if you want. So Dr. Leila Feiz, she got even 2 master's degrees. So one in cell and molecular biology from Tehran University in Iran, and a second one in plant science in Toulouse, France. And after this, she moved on to Montana State University to do her PhD in plant genetics. And by now she's working as a research associate in the Boyce Thompson Institute in Ithaca, where she's investigating plant protein degradation mechanisms with application in plant cell and developmental biology. But also in biotic and abiotic stress responses. So today, Leila will speak about F-box proteins that regulate growth and photosynthetic efficiency in maize. Yeah, very happy to leave you the stage.
Thank you, Margot. Can you hear me? Is that okay?
Yes.
Okay. Thanks everybody for coming to this webinar, and thanks to organizers for inviting me. Today, I'm going to talk about COI1, choronotin insensitive 1, which is an F-box protein and part of an E3 ligase. COI1 is known as a receptor for jasmonic acid in Arabidopsis and some other model plants. And today, I'm going to show that it might actually, it has adopted a different role in maize. As some members of the family have adopted such role. So jasmonic acid, or its active form, jasmonic isoleucine conjugate, is a lipid-derived phytohormone known as a defense hormone, but also it's involved in development. And it's made upon applying wound to plants, or when plant leaves is being chewed by herbivores. And when it initiates transcription factor of a cohort of genes that are involved in defense and development. The defense genes are against pests such as this, like violent fall armyworm, that can eradicate a cornfield in a matter of days. So jasmonic isoleucine is when the transcription pathway for jasmonic isoleucine is that it's sorry, can I have to take this, I cannot see this page. Yeah, I can't get rid of this. Anyway, so it's in the when jasmonic isoleucine is in low concentration, the main player in the transcription pathway is JAS transcription factor, JAS repressor, and they are a big family of proteins that sit on the MYC transcription factor and prevents transcription of MYC-induced genes, GA-induced genes. When jasmonic isoleucine is abundant, COI binds to this molecule, and this mediate binding to JAS, and so it brings it to the E3 ligase complex and polyubiquitinate JAS and sends it for degradation by 26S proteasome. So MYC is released to initiate the transcription of the genes that are involved in defense and development. Today, I need to talk about another phytohormone, which is gibberellic acid. And this is the hormone for growth and development. And the players are, the transcription system is very similar. The players are a little bit different. Here we have a repressor that's called DELA, and it's a repressor of GA-responsive genes, and it sits on the PIF transcription factor, prevents it from initiation of transcription. When GA goes up, binds to a receptor, and then this receptor mediates binding to an F-box, a different F-box protein, that brings the DELA to E3 ligase and sends it for degradation. And the PIF transcription factors are ready to release ready to transcribe the genes. So in literature, it is known that Arabidopsis seedlings, when they are treated with methyl jasmonate, which is converted to jasmonic isoleucine inside the plant, when they are treated with methyl jasmonate continuously, the plant and the seedling cannot grow. So there's always this trade-off between growth and defense, which is broken when in the COI1 single mutant of Arabidopsis. COI1 also has developmental defect in Arabidopsis and is male and sterile. So COI is basically just malic acid-induced genes are essential for pollen development as well. In monocots, 2 gene duplications led to creation of 3 COI genes. And we have COI2, which is essential, similar to Arabidopsis, is essential for pollen development, but a double cross of COI1 and formation, when researchers made a double cross of COI1, homozygote mutant of COI1, it turned out that it's longer than wild type, and it has elongated internodes. So because the defense initiation of defense genes was broken, the growth genes was started. So the mechanism turned out to be because DELA binds to JAS. When in the COI mutants, COI is not present to degrade DELA. So to degrade JAS. So JAS entraps DELA and removes it from the PIF transcription factors, so they can start initiating the growth genes, and the plants grow bigger, taller. In the wild type, when COI is present and it's under continuous JA isoleucine induction, JAS is degraded by COI, and DELA sits on the PIF transcription factors and prevents growth. So plants that are under wild type plants that are under continuous induction of JA isoleucine, they stay shorter. So we wanted to look at maize, and as I mentioned, maize is a monocot, and in monocots we have 2 gene duplications that led to creation of 3 groups of COI. We have COI2 separated from 2 chelates of COI1. All the monocots, such as rice, Cetaria, barley, they have 3 copies of COI. Maize has 6 copies because maize is an ancient tetraploid and didn't lose the duplicated copy of each COI during the evolution. So we decided, first I'm going to briefly mention a double cross of COI2. First, I decided to knock out COI2s, and I made plants that were homozygote for one COI2 and heterozygote for the other COI2. And when I self-pollinated them, I could not recover any homozygote double mutant COI2, and test crosses showed that COI2 double mutant is cause lethality in pollen and not egg. So it's the plants are not viable because of the pollen lethality. So going back to COI1, I made double mutants and quadruple mutants of COI1, and they were viable. And the first thing that I tested was to see if those violent caterpillars actually can grow better on this mutant, because remember that COI is assumed to be receptor for jasmanic acid, and it's initiating defense genes. So we assume that this mutant plant is compromised in defense. It turned out I didn't have the caterpillars didn't have significant mass increase on this mutant relative to wild type. And when we looked at the genes, defense genes that were differentially expressed in COI1, basically we didn't find that much. Most of the known defense genes were not changed between mutant and wild type. And we tested other caterpillar that was not specific to maize, and even the mass of caterpillars was reduced on the mutant. So this was interesting. We didn't have a compromised defense, but the seedlings, the plant had some developmental defects. While the first leaf and the second leaf of COI quadruple mutant was taller, were bigger than wild type, the third leaf started to have a growth delay. And this delay in this developmental defect persisted throughout the plant life until at maturity, it basically reached to the plant length was half of the wild type and the double mutants. And this was because of the shortened internodes. So the internodes were in the quadruple mutant was significantly shorter than the wild type and double mutants. Keep in mind that this is completely opposite of the rice mutant. So in rice, we had elongated internodes and longer plants. And so we first looked at the genes that were specifically downregulated in this COI mutant versus wild type and single mutants and double mutants. And the genes that were only knocked down in this mutant versus everything else were GA-induced genes, gibberellic acid-induced genes. And major groups of those genes were involved encoding proteins involving photosynthesis and carbohydrate and cell wall metabolism. Such as, and many of them were C4 specific genes, such as Rubisco, encoding Rubisco, and PPDK and other genes. Because we have these photosynthesis genes, and also because of these yellow stripes on the leaf of this quadrupole mutant, we measured elements, micro and macro elements on the leaf and plant leaf, and we saw that micro elements were deficient like they were reduced in quadrupole mutant relative to wild type and double mutants. Macro elements were not changed, and photosynthesis, two parameters that we measured, gas exchange and quantum yield of photosystem 2, both were significantly reduced in the mutant relative to wild type. So now we knew that we have a plant that is COI, which is a receptor for jasmonic acid, is gone, COI1, at least the quadrupole mutant of COI1, but gibberellic acid is initiation system is compromised. So repressor for gibberellic acid is DELA. We first looked at DELA, and as I expected, DELA was accumulating in the mutant, indicating that it's not degraded well. So the growth genes are not transcribing well. And so I co-expressed Maize DELA with alone in bentomiana and together with GFP and with COI. When it's expressed alone, it's going to nuclei very well. When it's expressed with free GFP, which also goes to nuclei, DELA is similar to when it's by itself. When it's expressed with COI, and this is the case for other COIs, it disappears. And when I apply proteasome inhibitor BTZ on the leaf, after a few hours, the part of the DELA came back to the nuclei. So indicating that this COI presence leads to DELA degradation, and it's proteasome inhibitor, proteasome dependent. So my current hypothesis is that COI2 is the canonical COI that binds to JAS, degrades JAS, and induces genes involved in defense. And specifically, we know that it's essential for pollen development and is expressed highly in pollen, COI2. But COI1, my hypothesis is that it somehow directly or indirectly leads to DELA degradation. And I think this is a C4 specific phenomenon because I have evidence from Setaria that knockout of the COI1 in Setaria also leads to similar phenotype of maize. So I think that because C4 plants evolve under harsh and arid conditions where jasmonic acid was constantly induced, they adopted part of their perception system, jasmonic acid perception system, to maintain their growth. So basically, the C4 plants broke this growth defense counteraction, this opposite effect on each other. And with that, I would like to thank a lot my co-authors and my collaborators, especially Kristen Xu, Shane, Wu, Kevin Ahern, and my boss, George Bender, for hosting me in his lab and for his extensive support. Thanks to BTI for hosting me for so long here, and the Plant Cell, and the funding from NSF and USDA.
Great, thank you very, very much for your talk. I think we still have a little bit of time for questions, so I can already see one in the chat, but still feel free to add some more questions that the author will be for sure happily answering also later on. So here I see the question, which method did you use to generate the double mutant of COI1 in maize? And if it's easy to generate double mutants in maize, or how long does it take to grow mutant maize plants?
Maybe I skipped that part because I was in rush. So the mutant the single mutants of COI1 were all made by DS insertion, which is a transposon insertion, and it's a single copy. And it's like you can move it and manipulate it in the plant. So that's the COIs, and COI2s were found in the mutant 2 uniform U were found from a collection. So the double crosses, you just pollinate them and keep genotyping until you find the homozygote plants. Sorry, what was the second part of the question?
How much time it takes to grow mutant maize plants?
I mean, it depends on the mutant. So if you are looking at a photosynthetic mutant that is lethal, it's a few weeks you get a mutant. I mean, they germinate after a few weeks, after 10 days, the maize seeds usually, and they survive for a few weeks and they die. But in this case, like COI1, for instance, it was similar to wild type. It's like a few months until it reaches to maturity. Depends on the weather, whether it's warmer, colder. You are growing them in summer or winter in the greenhouse.
Okay, thank you. Maybe a last short question, and then for the rest of questions, maybe you would move to the Q&A box to answer them. So did you also check for Zeta mice COI1s if they are functional in regulating the maize protein degradation?
Sorry, I couldn't understand that question. Can you?
If you did check if the maize COI1s are functional in regulating the Jess protein degradation.
The Jess protein, no,
Yes.
no, I did not.
Okay. Thank you very much again. And yeah, last but not least, I would like to introduce our last final speaker, which is Dr. Jiorgos Kourelis. And he held a PhD from the University of Oxford, and subsequently he worked as a postdoctoral researcher at the Sainsbury Lab in Norwich, UK. But now he moved on to become a lecturer in plant sciences and food security at the Imperial College of London, where he's basically focusing on fundamental questions in plant microbe interactions. And he's interested in understanding the function and evolution of plant immune receptors, and also in applying the obtained knowledge to bioengineer disease resistance genes. So today, Dr. Kourelis will introduce us to the bioengineering of secreted proteases and their conversion into extracellular immune co-receptors. So very much looking forward to our last talk. And yeah, the stage is yours.
Thank you very much, Margot. And let me share my screen. Here, going back. There we go. So thank you very much for giving me an opportunity to present this work, which is the last part of my PhD work, and I never actually had the chance to present this before. And before I start, I want to highlight all the names of the people involved. This research was done in the lab of Reinier van der Hoorn in Oxford, and I want to in particular highlight the contribution by many students. So I want to highlight Ollie, Ruby, and Sam, who were undergraduate students in Oxford, and Sonia, Parvinder, and Ana Lucia, who are master students and who helped on this project and made very important contributions. So when we talk about plants and we look at them in nature, most plants appear healthy. And this is because they have an immune system and they're actively resistant towards most pathogens. However, when disease does occur in rare and exceptional circumstances, this can have devastating consequences, in particular in an agricultural context. And these pathogens can be pathogenic and cause disease by virtue of secreting these molecules we call effectors. And they secrete these effector molecules both extracellularly in the apoplastic space, as well as intracellularly, where they really aim to modulate the host physiology and help the microbe colonize the plant. For example, by suppressing immune responses or by making the plant produce nutrients which are beneficial to the pathogen. And for the purpose of this talk, we're going to focus mostly on these extracellular space, these apoplastic space. And in this apoplastic space, many different pathogens, in this case, I'm highlighting Cladosporium fulvum, Phytophthora infestans, Pseudomonas, and Globigerina rostochiensis. All different types of pathogens have all converged on producing these small protease inhibitors that all target papain-like cysteine protease, making these PLCPs very important virulence targets. And one interesting case that I studied a lot is the case of Cladosporium fulvum, which secretes a small protease inhibitor AVR2. And this AVR2 binds to inhibits the activity of both the PLCP RCR3 as well as PIP1. And we know that in this case, PIP1 is the more abundant protease that appears to be more important in the interaction with the pathogen. However, in some cultivars, in some tomato cultivars, the interaction of RCR3 with AVR2 actually leads to an immune response, a hypersensitive cell death response. And this is because those cultivars have a leucine-rich repeat receptor-like protein, which sits here at the cell surface, and which presumably interacts with RCR3 to monitor the presence of AVR2. And when AVR2 is bound by RCR3, this leads to recruitment of the co-receptor SIRC3, and that leads to downstream signaling and ultimately immunity. And what we knew from previous research is that while RCR3 and PIP1 are found across all solanaceous genomes that we looked at, and they're actually paralogs and present in the same genomic locus, CF2 only arose in the Solanum genus. So we think that CF2 really evolved to co-opt an already pre-existing RCR3. And we'd previously looked at which RCR3 orthologs could be inhibited by AVR2. And that was quite a few of them, but this did not always correlate with the ones that could trigger a hypersensitive cell death response. So for example, the eggplant one here, while it can interact with and be inhibited by AVR2, it cannot trigger a hypersensitive cell death response together with CF2. And so we wanted to know what the molecular determinants are governing this interaction with CF2. And to do that, we first focused on the eggplant and tobacco orthologs of RCR3. And what we use in our experimental system relies on this transient expression in Nicotiana benthamiana, where we can take our receptors and effectors and variants, in this case of RCR3, and we can co-express this all together very nicely. And what we expect to see is that when we co-express CF2, AVR2, and an RCR3 variant that can recognize AVR2, we'd expect to see this hypersensitive cell death response in this sector that we infiltrate. And now, normally in nature, what would happen is that you'd have a fungal spore landing, and this would happen on microscopic scale. But because we make every cell produce this, we get this very macroscopic phenotype that's very easy to read out. Now, obviously, in the case that we have an RCR3 variant that doesn't interact with AVR2 or cannot interact with CF2, we would expect to see no response. Or it might be that in some cases, we might even have autoactive responses where in the absence of AVR2, there might still be a response. This is not something that we observed in this research. So when we looked at the eggplant RCR3, we first divided the protease in 6 different fragments, where we have a pro-domain that's cleaved off in the mature protease, and then we divided the rest in 6 arbitrary fragments. And we made all the combinations and tested which one could induce this hypersensitive cell death. And we found that one that contained fragment number 4 of RCR3 could induce this hypersensitive cell death response. And when we zoomed in, we found that there's one residue that is important for this, and that's this D244P mutation, that if we introduce that, now we get a hypersensitive cell death. And what's interesting about this is that when we look at the protease, and these are AlphaFold models of the protease, we have our substrate binding groove with the catalytic site over here, with the catalytic cysteine. But the residue that's now important for leading to this hypersensitive cell death response actually is towards the backside of the protein and not really interacting with what we think would be the site where AVR2 would interact with. Now, next we move to the Nicotiana orthologs of RCR3. And first what we did is, well, this ortholog is not inhibited by AVR2. And the way we look at that is we do this activity-based protein profiling, which essentially is like a Western where you only see a signal if there's activity. And when we apply an inhibitor, there would not be activity. And so we have our signal here showing that our proteases are active. Now, when we use a general papain-like cysteine protease inhibitor, we lose all of this signal. And now when we add AVR2 or an AVR2 mutant that is not functional with tobacco RCF3, we do not see any inhibition. But when we introduce one mutation that we knew was important for the interaction with AVR2, we see a reduced signal indicating that now this protease can interact with and be inhibited by AVR2. And at the same time, we realized that in all Nicotiana orthologs of RCF3, there were 3 residues that were missing. So we introduced those residues together with the residue to allow it to interact with AVR2. And now the nice thing is that only when we introduce both those mutations, we get this hypersensitive cell death response here. And again, when we look at models of the protease, we find that these residues, like the residue that interacts with AVR2 is near the substrate binding groove. But the residues that are required now for leading to hypersensitive cell death response are again towards the backside, and AlphaFold indicates that this would slightly displace this loop by introducing these extra residues. So what I've shown you thus far is that RCF3 orthologs can be quite easily engineered with minimal mutations to interact with CF2 and participate in AVR2 recognition. But for the next step of the research, we were wondering, could we now take this paralogous protease, PIP1, and can we bioengineer that to interact with CF2 and recognize AVR2? And in order to do this, we took a similar fragment swapping approach where we took our proteases, PIP1 and RCF3, with the prodomains which are cleaved off in the mature protease, and we divided this again in 6 parts, and we made all the possible combinations. And then we tested all of these out in quantitative HR assays. And we find that when we introduce 3 amino acid stretches from RCF3 into PIP1, these are fragment 3, 5, and 6, we now are able to make a PIP1 version that can trigger a cell death upon recognition of AVR2. And then moving on, and I'm going to skip a lot of mutants over here, but we made a lot of mutants to essentially come down to about 18 RCF3 specific residues that when we introduce these into PIP1 to make what we call PIP1 plus, we can now get this to interact with CF2 and recognize AVR2 very specifically. And this PIP1 plus is now 95% identical to PIP1, while it only shares 54% sequence identity with RCF3. And that's about as much as any PLCP shares in sequence identity with each other. And the interesting thing is, again, when we look at where these residues are on the models of the protease, we find that they tend to surround around the right-hand side lobe of the protein near the substrate binding groove and around the backside of the protein. And then we were wondering, okay, if this is the case, like, will this alter the substrate specificity? And in order to do this, in collaboration with Fatih Demir and Peter Hüsgen, we did a PIX assay where essentially you take a proteome, which you digest into peptides, and then you further digest those peptides using purified proteases. And then by doing comparative mass spec analysis, you can identify protease substrate specificity. And what we see here is the characteristic box plots for PLCPs, where they have the majority of their substrate preferences is in the P2 position. And we see that RCF3 has a preference for leucine, valine, methionine, proline, whereas PIP1 seems to have a stronger preference for phenylalanine and leucine. And then we further validated this using fluorescence peptide substrate assay, which is what Mariana did in the lab, Mariana Schuster, who was a postdoc at the time, and she recently started her own lab. And basically what we find is that indeed we can recapitulate that PIP1 prefers phenylalanine and this P2 position, whereas RCF3 prefers leucine. And now interestingly, our PIP1 plus mutant shows a substrate preference like RCF3. And when we went back to our model and the residues required for leading to HR, we realized that we inadvertently had mutated 2 other residues as well, which are near the S2 pocket. And we figured that perhaps we can mutate those back to the PIP1 residues and to create PIP1++. And this PIP1++ has a preference for both phenylalanine and leucine, so it's sort of in between PIP1 and RCF3. But it can also trigger this hypersensitive cell death response when co-expressed with CF2 and AVR2. And so basically what this research shows us is that we have this very interesting example here of where we have a protease that's become a co-receptor essentially. But in the absence of the receptor, you would expect to have neutral evolution on the interaction side with the receptor. And you would expect to get the protease to want to evolve away from being inhibited by the pathogen. Whereas in the presence of the receptor and the pathogen effector, you would expect the opposite, where you'd expect to get positive selection for interaction with the receptor, and perhaps also positive selection for interaction with these protease inhibitors. And basically what we did is, by decoy swapping, we think that perhaps we could do something similar in the future to what others have done with other examples where decoys are important for recognition of pathogen components, where in this case they took an NLR immune receptor, an intracellular immune receptor that interacts with a receptor-like kinase, or receptor-like cytoplasmic kinase, and it recognizes a effector from Pseudomonas. And by mutating a couple of residues in this receptor-like cytoplasmic kinase, they could swap the specificity. And the idea is that perhaps by swapping the decoy completely, we could also generate a system here, or systems like this, where we swap the specificity. And with that, I would like to thank all the people involved in the research. I would like to particularly thank Rainier, my PhD advisor, Sohin, my postdoc advisor, and also where I finished some of this research. Mariana, I mentioned, she started her own lab recently, all the students and the collaborators, Peter and Fatih for the PICS essay, and all my other collaborators. Thank you very much.
Thank you very much for the very interesting talks. So I can see a couple of questions in the box. So was the microorganisms identified on every level? I'm not really sure. Do you understand?
So in this case, well, I think we can say that like all of these pathogens, the protease inhibitors have been characterized and have been shown to target PLCPs. They also secrete other protease inhibitors that target other class of proteases. And I think making proteases just a very important molecular players in the apoplastic space, targeting pathogens for various reasons.
Okay, thank you. And there's another question I can see, if you could elaborate a bit more on what you mean by decoy.
Yeah, so in this particular case, we call RSO3 a decoy because when we knock it out, we don't really see an effect on the infection with Cladosporium fulvum. So it seems that PIP1 is where if you do knock that down, there's a very big effect on the infection by Cladosporium fulvum. So it seems that while RSO3 is targeted by AVR2, this is not the most important target of AVR2. And essentially what happened is when CF2 evolved, the receptor, it like now interaction with RSO3 with the pathogen effector actually tricks the pathogen into a recognition event. So in that sense, it's a decoy. But I think it's a bit semantic. Some people also use the word GARD.
All right. Thank you very much. Actually, I can so far not see any further questions. So I would really like to thank all the speakers of today's webinar once again. It was really great, very interesting talks. And I think I would like to hand over to Jason for the last couple of minutes, seconds.
Yeah, thank you, everyone. Thank you to our speaker, Margot and Ari, for this really insightful webinar. Thank you all to those of you who attended this webinar. As I've shared earlier, this webinar is recorded and will be made available later on. And so if you wanted to revisit and watch or pass it along to your colleagues, you're able to do that by sharing the link that we'll provide later on. And then additionally, as we shared, the link and this particular focus issue is already available online. So be sure to check it out, spread the word, and really dive into the content. But with that said, thank you all for your attendance, and we hope to see you into our next webinar. Have a great rest of your week, everyone. Thank you again, and goodbye.